welcome back to the classes again. So we finished in the last class about the protein structure and also the properties of protein. So now next we'll go on to the digestion and absorption of protein. Along with it, we should also know the abnormalities in digestion and absorption of protein. So when we talk about digestion and absorption of protein, we should know what are the dietary sources that we take in our diet, which is rich in proteins, right? So what do you think is the main source of proteins? The proteins that we take in our diet are mainly the cereals and the pulses. So these dietary proteins is the primary source of the nitrogen, which is metabolized by our body. So what is it? Around 70 to 100 grams of protein is taken in our diet. So the main function of proteins, we all know, it is for the amino acids is used for the synthesis of the proteins that are required for different functions in our body. And along with it, the carbon and the nitrogen atom, which we use for the synthesis of nitrogen, and also the non-nitrogen metabolites. Along with it, the carbon skeleton of the amino acids can be oxidized to yield energy as an energy source in our body. So now when we move on to the digestion of proteins, you should know the digestion that takes part in the different parts of the gastrointestinal tract, like the stomach, the mouth, the stomach, and then the intestine. So when we talk about the digestion in the mouth, the most important proteolytic enzymes breaking down here is breaking it down into their amino acids. But the enzymes are produced by different organs in the body, especially the stomach, the pancreas, and the small intestines. So as such, in the mouth, there is no digestion of the proteins. So next, we'll go on to what is the digestion in the stomach. So when the dietary source enters into the stomach, it stimulates the secretion of an hormone, which is gastrin. Now this gastrin stimulates the release of gastric juice containing hydrochloric acid and also certain other enzymes which are synthesized as zymogens. So zymogens are the inactive form of the enzymes which have to be converted into the active form. So the zymogens are pepsinogen and renin. So what is the function of hydrochloric acid and the pepsin? So what hydrochloric acid is? It denatures the protein, thereby it is hydrolyzes and provides the acidic medium in the stomach. And what is about pepsin? It's secreted as pepsinogen and it is activated into the active form by the hydrochloric acid that's present in the gastric juice. So then what happens? The pepsin cleaves the peptide bonds of the proteins like the aromatic amino acid, acidic amino acids, right? And also the renin. So what is renin? Renin is present in the infants and which helps the milk to clot. Okay. So now other than this renin, you should also know what is the importance of casein. Casein here is hydrolyzed to form the para-casein, which coagulates in the presence of calcium ions to form calcium para-caseinate, which is not curd, and then the pepsin will act. Okay. So this is the importance of casein and how it is hydrolyzed to para-casein in the stomach. So what happens next in the intestine? So in the intestine, the peptidases enzymes are secreted by the pancreas. So what are the peptidases? There are two types of peptidases, the endopeptidases and the exopeptidases. So the endopeptidases are like trypsin, chymotrypsin, elastase. These cleave the internal bonds on the peptides, okay, the polypeptides. So it breaks the internal bond. Whereas the exopeptidase hydrolyzes the terminal amino acids. So these exopeptidases are again divided into two types, the carboxypeptidase and the aminopeptidases. So these carboxypeptidases acts only on the C-terminal or the carboxyl terminal of the amino acid, whereas the aminopeptidases acts on the N-terminal or the amino terminal of the amino acid, and it is secreted by the mucosal cells. Okay, so there are two types of hexapeptidases. So the endopeptidases acts on trypsin and chymotrypsin and elastase, and it acts in the internal bonds to give a smaller peptides, whereas the exopeptidases hydrolyzes the ends of the peptides, and these are again into carboxypeptidases and aminopeptidases. So what is the next you have to know is the activation of pancreatic proenzymes occurs by the action of the enteropeptidases, right? So this enteropeptidases is then activated. Now what is the pancreatic juice? It is stimulated by cholecystokinin and pancreozymes. Now what is the optimum pH where these enzymes act is around 8, and it is provided by the bicarbonates which is present in the bile juice, right? So this is the digestion in the intestine. So now when we further talk about digestion in the intestine, the trypsinogen, which is the inactive form, the presence of enteropeptidases gets converted to trypsin. Now this activated trypsin acts on 3 other zymogens, that is a chymotrypsinogen, proelastase and procarboxypeptidases, okay, and gets converted into chymotrypsinogen gets converted into chymotrypsin, proelastase converts into elastase, procarboxypeptidases gets converted to carboxypeptidases. Okay, so this is the importance of trypsinogen getting converted into the active form and how it activates the 3 other enzymes. Now when we talk about this, you should also know that this is called as the peptide bond specificity. Okay, what do you mean by specificity? It acts on the particular amino acid. So what is it? The pepsin acts on the aromatic amino acids and the acidic amino acids. Trypsin acts on the carboxyl group formed by the lysine and the arginine amino acid. The chymotrypsin acts on the aromatic amino acid leucine and methionine. The elastase acts on the small non-polar amino acids like alanine, serine and glycine. The carboxypeptidases acts on the successive C terminals, and the aminopeptidases acts on the amino terminal. So this is known as a peptide bond specificity of the enzymes secreted by the digestion in the intestine for the proteins, right? So this is the specificity. So next, this is the digestion. Next we'll go on to the absorption of protein. So most of the amino acids mainly are absorbed in the small intestine, and it is released into the blood for transportation, and this is mainly about 2 mechanisms. Okay, so an energy is required for the amino acids to be actively transported by this. So you require sodium dependent amino acid transport, which are of 5 carriers of amino acids. Okay, so you have the small neutral amino acids, the large neutral amino acids, acidic amino acids, basic amino acids and cysteine, and then you have the immuno amino acids. So these are the 5 different carriers of amino acids. Okay, so when we talk about this, so next we'll go on to another one, the absorption of proteins. It is mainly by the MEISTER'S cycle, or we also call it as gamma glutamyl cycle. So this cycle is mainly, we see it in the intestine, kidney tubules and also in the brain. So what is it mainly? The amino acid, it reacts with glutathione. Glutathione is a tripeptide in the cell membrane forming gamma glutamyl amino acid. Now this amino acid is carried across the cell membrane attached to the gamma glutamate, and it is released into the cytosol. Now what happens to the gamma glutamyl and the dipeptide, that is the cysteine glycine moiety? It is reused in the synthesis of glutathione, and this is transported of one molecule of amino acid, and regeneration of glutathione requires 3 ATP molecules, right? So next we'll just have a look at the gamma glutamyl cycle or the MEISTER'S cycle. So as we said, the amino acid molecule joins with the gamma glutamyl transpeptidase, which is present in the cell membrane, and it is transported into the cytosol. Now what happens here? The gamma glutamyl amino acid is released, and it, in the presence of 5-oxoprolinase, gets converted into 5-oxyproline, and the amino acid is released into the cytosol. Now this oxyproline gets converted to glutamate. This glutamate again combines with the cysteine to form gamma glutamyl cysteine, and this gamma glutamyl cysteine joins with glycine to form the tripeptide glutathione. Now this glutathione again is resynthesized, and this whole process requires 3 ATPs, as you can see here, 1, 2 and 3 ATPs for the regeneration of the glutathione. So this is called as a gamma glutamyl cycle or the MEISTER'S cycle. Now when we talk about the absorption, you should also know what are the abnormalities of protein digestion and absorption. So what is it? There is defect in the pancreatic secretion, and it impairs the digestion of proteins, and it leads to loss of proteins in the feces. So these are the abnormalities which is generally seen in protein digestion and absorption. So the first most important one which we know is the Hartner's disease or neutral amino acid urea. So what is it here? There's an inability of the intestinal and the renal epithelial cells to absorb the neutral amino acids. So the tryptophan absorption is severely affected, and this leads to symptoms of pellagra. So we all know that tryptophan leads to the synthesis of niacin, and niacin deficiency leads to a condition which we call it as pellagra, which has characteristic features of 3D, that is diarrhea, dementia, dermatitis, okay, and if not treated will lead to death of the patient. So when you talk about Hartner's disease, you should also know something which we call it as deficiency of pancreatic secretions may be also seen in pancreatitis, cystic fibrosis, or also any surgical removal of the pancreas, okay. And in some conditions there is intracellular protein degradation by the lysosomes, okay. It can be like cathepase intracellular proteases, and then you have the ubiquitin intracellular protease, which is independent of the lysosomes, okay. So you have two intercellular protein degradation, which is one is lysosomal dependent and the other one is lysosomal independent, that is ubiquitin and cathepase. So these are the abnormalities of protein digestion and absorption. So in this you should remember mostly the Hartner's disease, right? And also pancreatitis, cystic fibrosis is also in another abnormality of protein digestion and absorption. So this finishes with the digestion and absorption of proteins. So in the next class we will go on to the general metabolic pathway of protein metabolism. So thank you and have a nice day.